Welcome, everybody, to this session on the Advanced Therapeutics panel on CNS. This panel is co-chaired by myself, Murali, and Paul. We have an excellent lineup of illustrious panelists today who will go ahead and introduce themselves in a few minutes. But what Paul and I have chosen to do in this session, this is an Advanced Therapeutics session, panel session, does not focus on any particular disease area per se. But we're going to cover some of the overarching themes and emerging trends that essentially cut across various disease indications. So we're going to cover essentially sort of four subtopics, if you will. One theme that's common is across biomarkers in CNS. We're going to cover that at the beginning. We'll move on to then gene therapies, which certainly a new modality that's opened up in CNS. Some of the progress and also the challenges or the controversies in that space, we'll cover that. Then we'll move on to sort of general considerations with respect to drug development in CNS. The challenges, some of the approaches that everybody is taking in terms of de-risking assets in the CNS space. And last but not least, we'll cover the commercialization aspects as well, particularly with respect to CNS drugs, neuropsych, in certain parts of the world, particularly in Europe as well. So what we'll do is, essentially go around. We'll start with introductions, and then we'll open up the panel session. So we will start with Paul first, and then we'll go around the panel, and I'll close with my introduction. Paul.
Great, thanks a lot. I'm Paul Matteis. I co-run the Therapeutics Research Group at Stifel and have a coverage focus and passion for neuroscience, as well as a few other non-oncology biotech areas.
Okay, let's go to Manuel.
Hi, this is Manuel López-Figueroa. I'm the managing director of Bay City Capital, a small venture capital firm in the Bay Area, specializing in healthcare investments, where we have funded and studied several companies, around 100. But I'm also scientifically a son of the Pritzker Neuropsychiatric Disorders Research Consortium that focus on psychiatric disorders. And I'm the founder, board member, and advisor of several biotech and venture companies.
Thank you. Go to Mark from Arvelle.
Thanks. Hi, I'm Mark. I'm the CEO of Arvelle. We're a company focused on bringing new treatments to patients with focal onset epilepsy. And our first drug should be able to hit the market in the middle of next year, 2021.
Great. Let's go to Michael from Metys.
Hi there, my name is Mike Scherz. I'm a chemist by training and founder of Metys Pharmaceuticals. We're developing MP101, a small orally active molecule for neuropathic pain. We have phase 2 clinical trials lined up for chemotherapy-induced peripheral neuropathy and diabetic peripheral neuropathy.
Thanks, Mike. We'll go to Parag then from Axovant.
Great, thanks, Murali. I'm Parag Meswani. I lead commercial strategy and operations at Axovant Gene Therapies. Brief background on me. I'm a pharmacist by background, have worked within neuroscience, neurodegeneration for the vast majority of my career, largely at Biogen, where I focused on MS, ALS, SMA, among other adjacent disease states. And more recently, I was at Spark Therapeutics, where I caught the gene therapy bug, or maybe I should say the gene therapy virus. And I'm now at Axovant Gene Therapies, where we have a pipeline of CNS-focused medicines, both on the rare monogenic disease side and pediatric lysosomal storage diseases, as well as in Parkinson's disease. So a very unique set of challenges and opportunities that we're advancing with the clinical programs that we have here.
Great, thanks. Sander, we'll close with you.
Yeah, Sander van der Heijden. I'm a partner at Forbion Capital Partners, and until recently I was the EVP of Research and Product Development at Unicure. I'm also a founder of Unicure, and over the years have developed several programs in the CNS space, including Sanfilippo, Parkinson's, but also more recently Huntington's disease and the SCAS, the spinal cerebellar ataxias.
Great. Thank you. And last but not least, I'll introduce myself. My name is Murali Gopalakrishnan. I'm with AbbVie, with the corporate strategy office here, with responsibility for neuroscience therapeutic area with respect to the external innovation. Scientist by training, a pharmacist and pharmacologist, been with the R&D organization for the last 2 decades or so in the company, in neuroscience being a major chunk of that time. And it's a real pleasure to be moderating this panel along with Paul. And this is going to be an exciting session, and we're going to start with the theme of biomarkers being sort of the first question here. It's a broad topic, and we can have multiple sessions and multiple days to discuss biomarkers. And it always comes down to something that's very important for CNS drug development, because, if you hear the word out there, it's one of the riskiest areas we are in terms of drug development and therapeutics. And translational strategy is a very important one with respect to any program. But with CNS, I think the question is around, we all gravitate towards biomarkers. So I think this is a theme that, cuts across pretty much, everyone here. And there's been considerable progress and evolution in this space, and there have been challenges. You hear a lot about, neurofilament lights, CSF markers, imaging modalities, but, it's a lot of places to go. But I think what will be important would be to sort of highlight what's been the compelling or the transformational events that's happened in this space with respect to biomarkers, and what are the challenges we see here. And maybe we'll start with, I guess, with Parag, if you could take us, because, kind of at the intersection of both gene therapy and neurology. Maybe we'll start there.
Yeah, happy to start off, and I'll give you maybe a couple of framing thoughts, and then I'll make it relevant specific to two programs that we have in development right now in Gm1 Gangliostitosis and in Parkinson's disease. But the reality is that the relative lack of biomarkers within the neurodegenerative space has traditionally been challenging, not just for genetic medicines, but across modalities. But I really think that outlook is changing. We're seeing a lot of advances in CSF biomarkers, in serum biomarkers, and capturing enzymatic activity. You have neuroimaging modalities like PET imaging that I think have come a really long way across different therapeutic areas, from Alzheimer's disease to how we're using it within our Parkinson's clinical development program. I think the challenge is within these diseases, especially for rapidly progressing diseases, you're still reliant on clinical measures just as outcomes, because the biomarkers may not be sensitive enough, or they may not give you a clear enough signal early on in drug development to be relevant. But I think that I'm optimistic, because I think that is changing. And I'll give you two examples for that are relevant for our program. One is for Gm1 gangliostitosis. So for those of you that don't know this disease, this is a pediatric lysosomal storage disease. It's monogenic in nature, caused by a single gene mutation. Fortunately, we have the benefit of naturally occurring models of disease. So there are cats and sheep that are born with Gm1 gangliostitosis, just like humans can be born with Gm1 gangliostitosis. So we're able to leverage and test these different biomarkers, both CSF markers of disease activity, enzymatic activity, and MRI imaging. And indeed, we've done so with partners at UMass Medical School and their gene therapy program there, along with a colony of these cats that actually live at Harvard University. And we've been able to evaluate the utility of these markers in preclinical studies. What we've shown with gene therapy so far is improvement in neuromotor function and extension of survival. And to put that in perspective, cats with these diseases typically are sacrificed at about 8 months. With gene therapy and with these early indicators using biomarker-based outcomes, they were a window to the future, because we now have evidence of these cats surviving out to 5+ years at this point. It's the biomarkers that gave us proof of concept. It's the biomarkers that give us a sense of safety and efficacy early on in the setting of no therapeutic options and in the setting of natural history, which is really well characterized for GM1. And so we're employing that model now in an ongoing clinical trial for GM1 that's going on here in the US at the NIH. That's the GM1 rare disease example. The one other one I'll briefly talk about is in Parkinson's disease. So we have a Parkinson's program that's in a phase 2 clinical trial right now. The gene therapy program that delivers 3 genes that are involved in the endogenous production of dopamine, right? This is the deficient biochemical that causes many of the Parkinsonian symptoms. What we're employing here in the clinic is PET imaging. PET imaging is a really great modality that us and a lot of other gene therapy companies that are evaluating PD approaches are using. And they're a measure of enzymatic activity. You can use it to measure AADC, which is one of the 3 enzymes involved in the endogenous production of dopamine. You can use it as a tracer to assess response and measure that effect within the CNS itself, and target activation ultimately as well. So this is something that we've incorporated into our clinical development strategy, and it's one that I think is going to be proved to be incredibly valuable as our program begins to read out more data later this year and into next.
It's great. Those great examples as we think about, gene therapy specific targets where there's good understanding of the biology that can take us in a compelling direction for biomarkers. Obviously, great examples there. Obviously we don't want to stop in neurodegeneration, but if you open up the dial a little bit more and think about neuropsych, I think that's a different world, right, as we think about translation and biomarkers. Maybe Manuel, do you have any thoughts that you could shed light in terms of how you think about this space, how you advise companies, and what is the recent trends? What are the recent trends?
Yeah, so you're absolutely right. I mean, psych is a different beast, and I remember attending several psychiatric meetings where the topic were biomarkers and are we there yet? And unfortunately, the answer is we are not there yet completely. Even though with the reduction of cost of genomics and, particularly, the ability of utilizing the gene expression profiling and looking at changes in gene expression in the blood or even in the brains of patients with psychiatric disorders, and combining that with other approaches like epigenomics or immune repertoire, metabolomics, or proteomics is advancing, and even to the level to single cell analysis. The truth of the matter is that, as far as I know, we don't have a truly validated biomarker for these heterogeneous populations. So the trend that I see, and is along the lines of our colleague was saying before, is the application of novel technologies or technologies that have been there before with a completely or a novel approach. For example, EEG, or using functional MRI to look at the connectome. In the case of the connectome, I mean, some of you may be familiar with the work of some of my colleagues at Stanford and Cornell, Nolan and Connor, where they have identified specific brain regions that can be used, for example, for non-invasive treatments, whether it's TMS. Saint study is one recently published one, where that's a good example of how you can tailor specific brain regions for the treatment with TMS. At the same time, there is new technologies in the digital space that is allowing to identify subpopulations of patients. And, it's the integration of all this information from the clinical information of the patient, molecular information, epidemiological information, that is allowing us to tailor and identify patients or patient subpopulations who can either respond better to treatments or even for the identification of novel targets. And if you want, I can go into details of some of these targets later on.
Thank you. Would anybody else like to chime in or add further here before I pass it on to Paul to jump into gene therapy again?
Well, we've had some recent experience with a surrogate marker in the setting of a monkey study that we've performed, where the monkeys have been treated with oxaliplatin. They develop cold sensitivity, just like humans do when they undergo their chemotherapy for treatment of their cancer. They also develop this first symptom of cold sensitivity. And in the monkey, of course, it's easy to measure the tail withdrawal time of these monkeys. Very straightforward. But our collaborators have gone to the trouble of putting these monkeys in a MRI machine. And I was stunned when I saw the result of in the lower section of the cortex, just like two thumbs sticking up, was this MRI signal. And that signal is responsive to effective therapy. So duloxetine, in this case, high doses of duloxetine, but not responsive to ineffective therapy, tramadol or pregabalin. And I was delighted when we saw MP101 work, not only on the tail withdrawal time, but also on this secondary marker, this MRI marker. But what are you going to do with that? It's nice. It feels good. It gives you confidence that what you've detected with the tail withdrawal time is relevant. But my goodness, it's expensive. It's slow. Would I use it to reduce the number of patients that we need to include in the study? Well, the trial that we've laid out, we know that 90% of the patients who take oxaliplatin develop sensory symptoms of peripheral neuropathy, 90% of them. So we'll run a prevention study. I don't want to exclude anybody from the study. We can treat everybody. They'll all benefit. So I don't need the biomarker for that. Am I going to run the study in just a handful of patients? Well, for our drug, we already have 200 patients worth of data from previous work, the compound. So there's no benefit for us to take that expensive biomarker and integrate it into what needs to be a very simple and straightforward question to the patient. Please score on a scale of 0 to 10 the sensitivity to touching cold items. Right? So the biomarker story is interesting. I was stunned to see this cold sensitivity light up so beautifully. But it is it's almost of academic interest.
But again, I think that brings up a very good point, Michael, because in some cases, some of these are very initial observations that you may not be able to immediately adapt to a trial, but maybe those learnings could help the next generation mechanisms coming forward.
That's certainly true.
So it's sort of like going back and forth in terms of bringing the learnings to innovate further as we think about the next line of therapeutics that is teaching us something from the first experiment. So it's really the cycle that we have to live through in translation. Let's kind of move on a little bit here, and I'm going to hand off to Paul, who's going to dive back into gene therapy. Paul, do you want to take it on from here?
Yeah, great, thank you. And I would love to tie it back to biomarkers in a second for gene therapy as it relates to maybe mechanism agnostic biomarkers, but maybe we can start with Sander, who's behind a lot in the CNS gene therapy space at uniQure. And that is, I'm coming at it from a different angle, right, on the investor side. But, one thing I hear from people looking at early stage CNS gene therapy assets is that there's a lot of existential risk when you try to vet a product, because there have been a number of setbacks and clinical holds. And for the drugs that look good, there's often not really that big of a safety database to kind of go off of, right? So to that point, Sander, maybe you can talk about your experience doing early stage preclinical and early clinical gene therapy development. And when you're kind of looking at early programs, how do you think about therapeutic index? How do you think about some of the issues with AAV9 and whether or not there's any read-through to anything else, right? How would you kind of advise an investor or a BD professional to kind of vet and get a handle on some of these kinds of things that sort of remain pretty salient questions? Yeah, do you want to start, Parag, and then maybe Sander can react to some of your commentary, which I think will kind of get at what I was asking about, which is really just making sense of the safety and therapeutic index of gene therapy in the brain and how a setback for one program does or does not read through onto another. But maybe, Parag, you can start with kind of your experience.
Yeah, I got a couple of thoughts here, and I'd love to hear Sander's perspective, given your legacy in the space. Look, what I would say at the outset is, what we know to be a fact, drug development is a risky business. The development of gene therapies is a risky business. And I think for us, what does that come down to? Which is gene therapy has come such a long way. And I think sometimes people assume that this is an overnight phenomenon, overnight success. Absolutely not true, as folks on this panel know. But we've come a long way. And the challenge is, how do you optimize vector design? How do you optimize vector delivery with the safe and effective dose? It's a similar challenge you would have for biologics and other molecules as well. How do you mitigate benefit risk in the context of what you can expect with a gene therapy? What we've seen over the past year, I think, are disease and product specific issues, in my opinion. The more recent example with Audentes of the MGM program, right? Incredibly high doses, but there are also some patient-specific risk factors involved along the way as well. And so I think there are lessons learned along the way about how do you marry the needs of the disease and the benefits and risks of treating a disease, in the case of MTM that doesn't have any effective treatment options, with the benefits you can deliver, finding the right patients that can benefit, and then delivering the safe and then the effective dose that's going to maximize the value of what a gene replacement or gene therapy can do. That's what we're trying to do here, right? So how do you mitigate that? What are some of the steps? Well, it's an iterative process, right? It goes back to understanding the biology of the disease in preclinical models. The biomarker piece certainly comes into play. Are there markers of progression? Are there markers of safety that can be employed early on in the process to ensure that you're doing the right things and you're not doing more harm than you are doing good? And so for those reasons, I don't think there's necessarily translation from asset to asset. But I think there's lessons learned, right? We're still learning about what good drug development looks like within the gene therapy landscape. And one company's success, it doesn't necessarily mean the same company, another company will have that same success, and vice versa. One company's issues for a program aren't necessarily transferable to others. We know the AAV9 can be successful. We've seen it. We've seen it with SMA, right? So how do you manage that and transfer the skills, the lessons learned from Zolgensma and Luxturna and perhaps even now hemophilia and some of the issues that regulators are now posing back to manufacturers, and take those lessons learned into good drug development for your programs. And so we're obviously students of the space right now and making sure we're taking some of those lessons learned and doing what I said at the outset, which is maximize the safe and effective dose with vector design and vector delivery and the right target tissue. That's kind of our strategy here.
Maybe I can ask Sander a follow-up. That's great. Thanks so much. And Sander, how do you think about, you go into the clinic, like when you were at Unicure with the Huntington's program, to what degree can you answer the safety question before you dose a patient, right? How good are animal models at vetting the TI or tox margins for these types of assets? Since we're living in a world now where preclinical gene therapy companies can come at a hefty price.
Sure. No, that's a very relevant question. And I think the only answer to your question I can give is that there's not a single animal model that will provide you with the answer. That has to do, of course, with the fact that depending on the technology that you use, you have to also look at human systems. For example, we've been working with silencing, and off-target effects for silencing in any animal model are of very relative value for humans. So what we've done in our development program for Huntington, for example, is that we have used 5 different animal models in 4 different species to spread the risk, so to say, and also to get more an idea about biodistribution and longevity of expression in larger animals, because I think it's in these instances really a stretch, so to say, to jump from a mouse to a human being. There must be something in between. And the primate might not be the best animal, because the primate's brain is, particularly when you use macaques, is basically as large as a cat. So the model that we took is pigs, because pigs have brains that are at least 10 times larger than that of a macaque. The other really important decision we made is that we should take look at human brains. So we set up a big organoid unit within Unicure, and so for each and every of these therapies, we have mini brains and organoids looking at efficacy, of course, and you can even do functional organoids now, so that's going very fast. And look at off targets in those models. I think those results are much more relevant than looking at off target effects, for example, in a murine model. So yeah, you have to combine all that. Now, the last thing we did is in animal models, we looked at functional imaging. So we did functional imaging in models that we thought were the best reflective of the human situation. And those exact imaging tests we are now using into the clinic. So I hope to get some comparison there, because we know that we can get some results on neural functioning or microglial activation and all that sort of stuff with functional imaging that in a one-to-one setting you can translate to humans. So that's the last part, I think, also. Getting back to the biomarkers, to have biomarkers that we can move from preclinical to clinical.
Yeah. And what do you think about, we talked about biomarkers across a few different areas, but there's always a lot of discussion around drug mechanism-specific biomarkers in gene therapy showing you get into the brain, maybe you have a substrate, but what are yours? And I'd be curious where your head is at too, Parag, and in Parkinson's, on some of the kind of drug mechanism agnostic biomarkers like neurofilament and MRI, things like that. Like, are those ready for prime time to de-risk a drug on a small sample of patients, or are they still somewhat exploratory?
Are you asking the question?
Yeah, you go ahead, please.
Well, I can start off. So I think, the global biomarkers that give you an idea of damage or atrophy, when you talk about a disease that is, characterized by atrophy, we do know that atrophy, for example, in Huntington's disease, is by far the best predictor of survival and lifespan. So biomarkers that give you something about atrophy would be fine there. If you look at biomarkers that are thought, by many people to be very specific for that disease, like mutant Huntington, I think there is a problem, because the problem, of course, is where does that mutant Huntington come from? And there's a big discussion, about the validity of mutant Huntington lowering in the CSF. First of all, we know that such biomarkers are really relatively poor reflective of what's going on in cells. And then they're even poorer when it comes to showing or being a marker of what's going on in deep brain structures. So for those biomarkers, actually, I think it's interesting to see those. I think they will, in the end, be not very predictive, and I would rely far more on functional imaging, because that would tell you really what's going on for example, in Huntington in the deep brain, or for the SCAs in the cerebellum and the brainstem. I would be much more interested in that. But Parag, maybe that's my 5 cents.
Yeah, I share that perspective, certainly in the case of Parkinson's disease. I don't think that neurofilament light is really ready for prime time just yet, but I do think that imaging is coming into a new position, and certainly we're trying to advance that through our clinical program. To make it specific, with PET imaging, scan on the outside tells you what's going on the inside, right? It's as simple as that. And I think for us, with our Parkinson's gene therapy program, the question is, can you decrease the fluctuation, the variability that occurs with pulsatile dopamine in the off and the on states? And you can measure this, right? You can measure this with PET imaging by comparing the difference in different ligands and different tracers, and look at that change at baseline versus post-gene therapy administration. We're doing it. So we'll know the viability of that, at least for our program. That's come a long way, and I think it's something that we're really excited about. I contrast that again, I think, with the rare disease side, where there are different exploratory markers, toxic protein accumulations that we're measuring in the CSF, that we're measuring within the serum, enzymatic activity that we're able to measure in the CSF and also in the serum, and some other assessments as well, that are more exploratory. I think we can take more risks there and be a bit more creative in what we look at on the rare disease side. I don't think we have that luxury on the Parkinson's side, where we are focusing on more tried and true measures of therapeutic response, both clinical measures as well as imaging-based measures as well.
Yeah, great. And maybe one last gene therapy question before we switch to the next topic around trial design and patient selection, where we can get a few others to opine. But maybe I'll go back to Sandra and then hear from you again, Parag. And that is just, what do you think the delivery of gene therapy in the CNS looks like in 10 to 20 years? Right now it's highly invasive, often surgical or ICM, maybe intrathecal, but less success with biodistribution that way. It feels like when these programs go into the clinic, they often go right into very, very small samples of patients. And oftentimes FDA even pushes you more towards the moderate end of the spectrum where you'd love to treat earlier. So what's kind of doable for innovation on the delivery side that can kind of broaden the addressable patient population for these medicines?
Yeah.
Do you want to start, Sandra?
Yeah, so, the current status is that, with the best delivery systems, you probably hit something like 30% at most of the target cells, if you look at deep brain structures or the putamen, or, particularly with lysosomal storage disease, the complete brain maybe. So that is the limit, the best vectors hit the minority of cells. And when we talk about neurodegenerative diseases, that is a problem, because if you hit 30%, you save them, but 70% still dies. That's an issue. Yep. So the solution right now, for example, for Huntington that we worked towards, is to have secondary spread of the silencing mechanism by exosomes, for example, with microRNAs, and we have real hard data now to show that the cells around the primary transfected cells actually are also silenced, because there is exosomal spread of that microRNA. I think going to the future, clearly we need an improvement on vector systems, and this is one of the things I'm currently really focusing on. And that would entail either viral or non-viral systems, where you could target specifically cell types. So I'm now talking about neurons, microglia, astrocytes, microglia is a no-go territory right now for gene therapies, and so are Purkinje cells, for example, where there's a real need to get there. So there are technologies now in AAV, capsid technologies that could get you there, or non-viral technologies. So this is really the era of multi-specific drugs. Drugs that both have an inbuilt targeting compound and an efficacy compound, and I think that's why this next decade is going to be so interesting, and why the next decade of drug development really is going to be the CNS, as far as I'm concerned, because this sort of drug is going to solve quite a lot of these issues. So I'm pretty optimistic, actually, that within a few years we'll get away from either very high dose systemic AAV9, which can lead to problems, as you know, or intrathecal or intercisternal, or definitely interparenchymal administration going. Probably the next phase will be further development of intercisternal or lumbar intrathecal administration. But I think there will be a next wave after that of targeting far more specifically neurons, astrocytes, whatever cells you want, motor neurons, dopaminergic neurons, and so on.
Right.
The other thing is promoters, because there is a big development in promoters, and those promoters also are, cell specific, so motor neuron specific, dopaminergic specific. So that's another one that's going to help quite a lot there.
I think it's quite useful to keep in perspective that what small molecules do and have been doing for a long time have solved all of these problems. If you listen to what Arvelle are doing with cenobamate, or what we're doing with MP101, all of these issues just are solved. They're just solved.
Yeah, but
and that's a beautiful thing.
Yeah.
I admire the courage that people and the energy that are being put into these genetic-based therapies. But I have to tell you, my goodness, what a long shot.
Well, there may be a happy marriage here where you have tunable systems that you can tune with small molecules in some ways, right? So you can marry the benefits of both. So.
Yeah. It's it takes a lot of courage to try and think about genetic-based therapies for neurodegenerative processes where the delivery of the vector can't get to where it's needed. Small molecules do.
Well, if I may push back a little bit there, we spend all this time, to show that we are targeting actually where we need to target. So in that respect, currently gene therapies are, I would say, superior to, let's say, antisense technologies where you are completely right?
Yeah. And I would also add, I used to work in the immuno-oncology area, as far back as 1985. And it really took until 2006 to actually begin to see the right immuno-oncology agent, Yervoy, and the right kind of responses that made it therapeutically acceptable.
Yeah.
Then you fast forward another 10 or 15 years, and it's been super optimized and become such a mainstay of therapy. And I think gene therapy is going to follow, hopefully, a similar kind of path. I really encourage those to persevere, because I think at the end of the day, we'll figure out these issues as we have in other categories.
Great. Murali, should we move on? Oh, wait, do you want to lead off the next topic on drug development and mitigating clinical risk?
Well, let's continue on. Paul, if you want to continue on, that's fine. Do you want?
All right. Well, I want to make sure we have time to kind of involve everyone and everyone's interest. So maybe we can switch gears and I can ask a question to you, Manuel. Maybe you can talk a little bit about some of the work you're doing on the neuropsych side as it relates to kind of how you're thinking about trial design, patient selection, all those sorts of issues where maybe we'll, even in an area like psych where you might know a drug is active, it's all about how you ask the question and how you enrich the population, et cetera. So you can take this wherever you'd like to go, and then we can always kind of follow up and open it up.
Yeah. So I may take a tangential on that because I'm involved in a company called Ividomics, which is basically trying to help biotech and pharmaceutical companies to develop clinical trials in the immune-mediated inflammatory diseases. And the point on saying that is that the goal is to integrate high-quality clinical, epidemiological, molecular information in order to identify which subpopulations of patients will be amenable to a clinical trial, for example. That's one of the uses of that. And the same applies for psychiatry. The issue there is that historically we have not been able to, as in oncology, for example, in other indications, to get access to highly characterized, high-quality clinical information. We're starting to get molecular information. The Pritzker Consortium, for example, works on samples from postmortem tissue and also live patients. But as you can imagine, postmortem tissue have limitations and it's not very applicable to clinical trials. However, the identification of, again, novel targets like the fibroblast growth factor and the signature in the blood of those same metabolites, FGF related, allows for the identification of patient populations in, let's say, depression that hopefully will be more amenable to drug treatment. So that's what I'm seeing now. And as I mentioned before, we also see a lot of more of non-invasive technologies, whether it's TMS, whether it is the utilization of, for example, auricular vagal nerve stimulation through the ear that have been used in different psychiatric disorders, whether it's TRD or postpartum depression, and how you can, again, by means of the, for example, functional MRI, they start to identify those patients who have certain profiles that will be more amenable for that treatment. So again, I see it in the context of my colleagues at Stanford using the, as I mentioned before, the same study or the studies that we are conducting in an academic setting, as well as some of the companies that we are funding through the city capital.
Very good. Michael, do you want to comment on your experience as it relates to kind of managing the exogenous variables that can lead a trial to potentially fail, even if the drug works, and how it's relevant to the indications you're focused on?
Most of the work that we've been doing has been following the recommendations that were developed by the FDA. And about 10 years ago, 12 years ago, they began worrying about the high failure rate of studies in neuropathic pain. And clearly the drugs were known to be effective, but you couldn't figure out why. And that the trials were failing. Why? So it's not the drug that fails, it's the trial design, it's the questions that we ask the patients. And there's a whole series of conversations in this FDA guidance, consortium of FDA and academic and industrial sponsors, patient organizations as well. There's a lot to be said about training, and training not just the staff, but training the patients as they come into the study. But perhaps one of the most important things that we've focused on is to change our trial design. In diabetic peripheral neuropathy, where there are already several approved drugs, we want to run a clinical trial where we will randomize to placebo or to active treatment only those patients who've previously been responsive to the drug, to our drug. And by this selection process, wind up with a higher response rate. We know that the people who have entered the study actually do respond to the drug. So there's an open label run-in period where we ask if your symptoms have gotten better. And that, if you will, selection process then gives you a patient population that you know are responsive to your drug. Now you come and randomize those patients to continue on active drug or to placebo, and you see the placebo effect wash in. It's a randomized withdrawal design that's been effective in phase 3 trials in schizophrenia and depression and Parkinson's as well. So a randomized withdrawal design. So there are a number of things like this that can be done to increase the sensitivity of the assay. At the end of the day, though, the only thing that matters is that the trials are very carefully done. There's no substitute for taking care in the way that you prepare the patients, the way you prepare the staff, the way you run the trial. You can't do it just on the quick.
Got it. Okay, very good. Anything else to add on trial design, mitigating risk? Anyone else want to opine?
No, the only further comment is on the utilization of more digital health tools and how those are starting to be integrated. Obviously not as primary endpoints, but as a way of collecting data that, again, allows you to identify respondent or responders.
And compliance. And compliance. Compliance is important.
Manuel, what do you think is the kind of future of those data tools from a regulatory perspective? Like, do you think, like, there's a lot of talk on Insight identifying a certain blueprint of patients that respond to this drug, and it all sounds great, but do you think we're really going to get to a point in the near term where we can codify a homogeneous population from this that is prospectively recruited and then validated by FDA for a drug approval? I've met some of these companies and it's felt a little bit more academic. So when do you think this is going to be actually kind of truly part of a drug approval process? How do we get there?
I think that in addition to not just the regulatory aspect, I mean, you have to also ask the question of the payer's perspective on this and the value creation. So are we there yet? I mean, as you all well know, I mean, you have companies like Pear, Click just announced a big deal with BI this week, and then you have Akili approval. I mean, there is a number of these digital health technologies that have been approved or in the process of being approved or have received significant milestones or payments in partnerships. I think that going back to this idea of integration of data, I mean, quality of data is going to allow us to be more precise on the utilization of those in the selection of patients. And that's a matter of time. Is it going to be 2, 5, 10 years? I think I think the jury is still out there. But it's clear that that's a direction that we're going to be taking, especially now with COVID, where we know that telehealth and the ability of utilizing, CBT, cognitive behavioral therapy, or similar approaches to treat the patient, so to evaluate the patient, is going to be the direction to go. So you have seen the FDA being more lenient and allowing certain technologies, whether it's related to treatment or even diagnosis in psychiatry or other indications, to move forward in the last few months. Will that change when we have, treatments for COVID? I mean, it's still to remain to be seen, but certainly there have been an advance of these technologies, and the more data and the more validation that we're able to get, that would be the way to go. It is tricky because at the same time, you're getting a number of technologies out there that is very difficult to differentiate who are going to be the leading one. And in most cases, it's the business model associated with those companies that is the tricky part. And we can go into more details if you want on that.
Yeah, right. Okay, very good.
Maybe just a comment here, Paul. I think we're not going to talk about COVID and the impact on CNS drug development in this session, but I think just coming back to Manuel's point on the impact of COVID-19 on how we think about digital tools and technologies sort of forcing you to kind of operate in that way will probably shake out the most promising ones to be more mainstream as opposed to still keeping in the academic world. So that may be a true test of how they're going to perform with respect to CNS therapeutics.
Yeah, makes sense. I
think we probably have another 8 minutes or 7 minutes, depending on how you count them. We should probably shift to the last topic, right, Paul?
Yeah, absolutely. Do you want to kick it off? And
maybe start it off. And I know we touched on some of these early on as we think about So the ultimate objective of what we do is, making an impact to patients, and commercialization becomes a real driver on how we do that. And, this probably, maybe Mark can start off with it as we think about CNS broadly, and then particular challenges with respect to neuropsychiatric drugs. And, this being a conference with respect to biotech in Europe, certainly there are challenges as we think about neuropsych drugs and launching in Europe. So I don't know, Mark, maybe you would start off with a high-level perspective from your side, and then perhaps dive into the implications as we think about European market and other markets as you launch and, get into a product in the marketplace.
Sure. And probably just a couple of framing comments that I would make to start off the conversation is, the first thing that I think is critically important is you have to have a difference in your product, and you got to be able to communicate that difference effectively, and that difference needs to ultimately be meaningful to the clinicians and the patients. And a couple of examples that I've lived through is, in the antipsychotic area, we ended up having a drug that was comparably effective in aripiprazole, but didn't have the weight gain, didn't have the metabolic side effects. And ultimately, we had to make that an important issue that previously hadn't been talked about. As it relates to epilepsy and what we're dealing with now, the key unmet need is efficacy, because 40% of patients, despite a couple dozen medications, remain seizing even after multiple different trials and failures. And therefore, being able to help physicians understand that this is a different drug in cenobamate compared to all their other choices, and many of them that they're quite familiar with and in lots of cases have had success with, is going to be really, really important. And then when it comes to reimbursement, obviously the difference between the U.S. and Europe are dramatic. And I think it's particularly in CNS, where a lot of the trials are against placebo. It's really going to be very hard to have new drugs in Europe compared to the U.S. And I think in Europe, it's going to be much more on the side of drugs where you really are confident in your effect size difference, or you go after the rarer, more niche subpopulations.
Hey, Mark, can I ask, I'd love to get your thoughts on one thing. If you look at CNS in for emerging biotechs, Alkermes, Neurocrine, Acadia, they've all just kind of, without saying it, feels like they've kind of determined, what, Europe might be an NPV negative for us. So we'll just kind of bail. And if someone acquires us and wants to do it, great. What do you think is going to take for you guys at Arvelle to kind of buck that trend and really drive like a great European launch?
So I think the difference is I've worked on dozens of products, and this is the one that has the greatest amount of differentiation against the market's greatest unmet need. So I think in the work we've done, the penetration you get into the market is much more significant than lots of other products I've worked on with less differentiation. The other thing is treatment referral patterns for refractory epilepsy in Europe are well defined. And so, you really need to call on a smaller set of hospitals to get to where these patients are treated. And so you don't need a large commercial footprint. That, with essentially much lower prices than you could get in the US, still allows you to have a P&L that works. And, I've again, like many of the folks on the call, I've worked on many products where the P&L doesn't work unless you're able to be the second or third product call and then, share the resources. So I think, we're fortunately in a unique position where we've got an asset that clearly has that differentiation that makes it all work.
Yeah, that's really an important consideration. And just coming back to Paul's comments, from a BD perspective, especially in neuropsych, certainly several companies approach the big pharma, and the initial position is we would like to keep the US. You can deal with Europe, and that usually is a challenging place to start, really, because of all the right reasons you mentioned, Mark. How do we get a transaction going in that framework? That becomes really a challenge for us.
Yeah, and even on the investor side, as we're out talking to investors, there's a lot of investors who just find, one, they don't often like launches, and two, they often don't like launches in Europe because of the complexity where you got to go market by market to understand what kind of pricing you can get. So it even makes the capital raising a bit more challenging.
Exactly. And maybe we'll come back quickly to gene therapy and sort of commercialization in that space. I mean, that's continued to evolve, and certainly there are challenges in that space with respect to pricing and what the payers are willing to pay, for example. I don't know, Sandra, do you have any comments based on your view of this space now and going into the future?
Yeah, so for now, some gene therapies actually are cost effective. So if you look at the hemophilias, for example, if you get the longevity that we had with our hemophilia B program, basically, the longest patients out there are now, 9 years, and in our own programs, beyond 5 years. So if you just compare that with the cost of replacement therapy, it's cost effective. So longevity of expression is a big one. And I think for gene therapies, my own goal is, I wouldn't develop anything that doesn't express at least, in a therapeutic manner for less than 5 years. I wouldn't go for that because I don't think it is worth the while. Then when it comes to the cost of treatment for gene therapies, a lot of it is driven by very high cost of goods. And I think that we're going to see a major change there. So I'm talking about, cost of goods going below $1,000 or something like that for gene therapy for the brain. So these are critically dependent on, major improvements in process development and manufacturing. So that's what's going on right now. Gene therapies are not staying, the very expensive $1 million therapies that they are now. Definitely are going to get away from that. And then, the last thing is that, talking about gene therapies and small molecules and all that, there are just basically many neurodegenerative diseases where it's hard to target, with a small molecule. For example, when you talk about aggregation or prion-like spread and those sort of things, or when people, some companies are taking approaches by giving systemically administered antibodies, and they don't penetrate the brain very well, and so on and so forth. So their gene therapies could come in, and for Huntington's or the SCAs or whatever, there is no alternative. So if you come with a therapy for Huntington's, I'm pretty sure you get a decent reimbursement also in Europe, because there is no alternative, and it's a dreadful and deadly disease. So that's where we're going to probably start, and then going forward, as the therapies are going to be easier to administer and much cheaper, I think many neurodegenerative diseases are going to follow.
Yeah. Parag, would you like to add anything more to that with your commercial hat, thinking about Europe, for example?
Yeah, the only thing I would add is that maybe I'll stick with this market access and kind of price and access theme. We joke about this, but it's Star Wars medicine and a Flintstones healthcare system. Maybe it's a U.S. specific analogy, but I think it still holds true. I think for us, the challenge with gene therapy, provided we make the progress that Sanders is alluding to in the future, is how are you going to pay for it? What are the payment models that need to be implemented? And I think we're really, really early on in that process. I think here in the U.S. with the Luxturna and the Zolgensma experience, starting to scratch the surface in terms of outcomes-based models and thinking about annuity-based payments and starting to get creative. A lot more work needs to be done at a system-wide level, not just from a manufacturer point of view or from a payer point of view or a provider point of view. All stakeholders. Everybody needs to be on the same page to make this innovative medicine make it to patients. And I think if it's just from a singular manufacturer vantage point, it's not going to be successful. It really has to be all hands on deck to really come up with creative solutions if you want promising science to ultimately make it into the patients that need it. And I think we all owe it to patients. We have that obligation to do that well and to be proactive in how we think about that early in the drug development process.
Got it.
Yeah. And just one point about, in terms of in the field of medical devices, earlier we sought Europe as the way to go and, go through the regulatory agencies and get into the market, get adoption and feedback from the users. At this point, that's changed. Therefore, at least when it comes to medical devices, not necessarily the case. And there's more parity between the US and Europe. And the other topic, I mean, you guys have talked is, many of these indications are orphan indications, and the payer's perspective on how much they're going to pay is going to be interesting moving forward. And there's a big difference between, obviously, the US and Europe.
Great. All right. Well, Paul, I've been told that our time is a couple of minutes, maybe 2 minutes left. Do you have a final question or thought, Paul, from your side that the audience or the panelists would like to answer, or do we want to close it out? Paul?
I think we can close it out. I think it's hard in this area to ask any open-ended question and end it in 2 minutes. Look, I thought this was a great discussion. We covered pieces of a lot. Anything else? I mean, I guess, Murali, you do a lot of looking at companies and different assets, right, on the AbbVie side. How do you think about balancing risk and upside, right, for some of the emerging science and kind of clinical trial existential risks when you make recommendations to your higher ups on where to deploy capital? Maybe we can end it with that. I'm curious.
Yeah, no, I think it's that's not a direct answer, and it's not going to be a quick 1 minute answer as well. It's going to be case by case. Obviously, one thing we focus is good science. I think once we narrow down into really compelling science that can be translated from a hypothesis or an idea to a human experiment, I think that trumps everything, and then everything else follows from that. So irrespective of early stage or late stage, I think really locking down on the good science and confidence in that data is really the key driver for us.
Got it. Okay. Very good.
Great.
Well, thank you everybody for taking the time. It was interesting to hear everyone's perspective.
Thank you all. It's been a great session. Look forward to seeing you all in person one day.
Gladly.
Yeah.
Hope so.
Thanks.